Alright guys, let's get busy talking about germ layers, which are incredibly high yield on step 1. Let's start with an overview of what we're going to cover in this video. So starting around week 3, we have a process that is called gastrulation, and this is the process by which we have formation of our 3 germ layers. So what happens is we have our epiblast, which is this blue structure here on top, and it is going to proliferate and migrate to form 3 different germ layers. And so our germ layers, starting with the most superficial, is going to be ectoderm, and then if we go deeper, we're going to have our mesoderm, and then if we go even deeper, we're going to have endoderm. And it is these 3 layers that we're going to talk about in this video and the things that they go on to create further down in development. Let's start off our discussion by talking about ectoderm, and when it comes to ectoderm, let's reference our original guiding model. So we have our ectoderm here, which is the most superficial layer, and when it comes to the ectoderm, I like to think of the ectoderm as the attractoderm. And I like to call it the attractoderm because things that make you attractive are your beauty and your brains. So we're going to talk about both beauty and brains and the structures related to them and the ectoderm. So in terms of our beauty, it's going to come from our surface ectoderm. So we have our epidermis, which is our skin. We have our hair, our nails, and our salivary glands. And then we're also going to have our adenohypophysis, which is going to turn into our anterior pituitary. And our anterior pituitary is going to make hormones, and we know that hormones are responsible for giving us our good looks in puberty, and so that helps me remember that this is going to be derived from a portion of the ectoderm. Let's move on now to talk about brains, and this is going to be all about neuroectoderm. So from our neuroectoderm, we're going to get our central nervous system, and that's going to include our brain and our spinal cord. And this is really high yield. They love to ask about this on test days, so let's go into some of the specifics of the embryology. So if we look here, this entire area that I'm kind of shading in blue here is going to be our neuroectoderm. It's going to include this green and this purple population of cells. So specifically, we have a population of cells called the neural plate border, which will become important a little bit later in our discussion. And then we also have our neural plate. So let's talk about what happens with these populations. So these cell types are going to undergo a process called convergence. And essentially what's happening is they're going to fold inwards and upwards, and they're going to create this structure called a neural fold. At the same time as this kind of folding action is happening, we're going to have an invagination below, and this is going to be called the neural groove. And so these 2 structures are kind of forming at the same time. And additionally, as this is happening, this blue layer of cells is going to form the overlying epidermis. So at the end of this process, we have our overlying epidermis and we have our neural tube beneath. And the neural tube is going to be a really important structure because the neural tube is what forms our brain in our cranial regions, and then our spinal cord more caudally. And it's going to be important in terms of pathology. I want to mention that during this time, both of these structures are really reliant on folic acid levels. Folic acid levels are super important for neural tube closure. So when patients don't get enough folic acid during pregnancy, they are going to have fetuses that have neural tube defects. A neural tube defect we're pointing out here is called anencephaly, and you will notice that this fetus is missing a portion of its brain, skull, and overlying scalp, and this is because it had failure of neural tube formation in this area. We can also have failure of neural tube formation further caudally, and when this occurs, we call this spina bifida, and this is when it is the spine portion of the neural tube that does not close appropriately. So let's just do a quick high yield review that it is our brain and our spinal cord that are going to form from our neuroectoderm, and it's going to be highly dependent on folic acid levels during early months of pregnancy. Let's move on now to talk about the next subset of cells that is going to be really important that is actually derived from the ectoderm, and I didn't mention it initially because I didn't want to get too complicated, but now it's time to insert it in our overarching diagram. So you'll notice we have these little green cells down here that I'm highlighting, and these represent neurocrest cells, and they are a portion of the overlying ectoderm. So they are a subset of ectoderm, and they are a very special subset that the test loves to ask questions about. So these are actually going to come from the neural plate border, which we talked about earlier, which was that special little green portion, and we can follow this down through our diagram and see that they become a distinct cell population down here in our third image, and they're going to go ahead and migrate throughout the rest of the body, and they're going to form specific structures that are very high yield. So let's go over some of those structures. So we're going to have neurons of the peripheral nervous system as well as the leptomeninges, and then our neural crest cells are also going to form the bones and connective tissue of the skull. Specifically, high yield here is going to be odontoblasts, which are going to be the precursors to our teeth. Additionally, if we move down into the mediastinum, we're going to have the endocardial cushions and the aorticopulmonary septum, which form from the neural crest cells. And then if we move into our skin, we're going to see that we have melanocytes from our neural crest. Now, some additional cell types are the chromaffin cells of the adrenal medulla, as well as the enterochromaffin cells, which we find in our gut lining. Let's move on now to talk about our mesoderm, and in our overarching guiding structure, we'll see that our mesoderm is this mid-level structure that was formed upon gastrulation. Let's talk about some structures that are important for mesoderm. So in terms of our axial mesoderm, it's going to create our notochord, and our notochord is going to go on to form our nucleus pulposus, which is kind of like the meaty portion of our spinal cord that is going to be responsible for disc herniation syndrome. So when you have a patient with back pain and a disc herniated, this is going to actually be the nucleus pulposus, which is a part of mesoderm that has herniated out. Next, let's talk about a paraxial mesoderm, which is going to be all about musculoskeletal. So it's going to be the vertebrae, the ribs, and our skeletal muscles. Next, importantly for this unit, we're going to have our intermediate mesoderm, which is going to be all about the kidney and the gonads. So we will be talking about this a lot in our unit. And then finally, we have our lateral plate mesoderm, which is going to be responsible for creating things like the cardiovascular system, also our microglia, our stem cells of hematopoietic origin, and our limbs. Now, you may be asking, why are microglia a part of our mesoderm? Because aren't microglia part of like the CNS and the neurotype structures? And this is a great, fantastic question. However, I like to think of microglia as like the macrophages of the CNS. And so if we remember, our macrophages are actually going to be of hematopoietic origin. And so it's these microglia that actually come from this hematopoietic origin as like a CNS macrophage, which is why they're considered a mesoderm. And that's a tricky little sticking point that test writers like to ask about. Let's wrap up our discussion by talking about endoderm, which is the third part that is created in gastrulation. It's going to be the deepest structure in our overarching diagram. And when it comes to endoderm, I like to think of the most innard structures. These are going to be our organs that are lined with epithelium. So when it comes to our organs and the epithelium, I think GI tract, respiratory tract, urinary tract, these all have complex epithelium that are responsible for epithelium-like functions like gas exchange and transport of nutrients and movement of urine throughout our system. So that's how I like to think of endoderm. I like to think of endoderm as classically like the guts of the body. So esophagus, liver, GI tract, intestines, those kind of things. But additionally, our endoderm is also going to be found heavily throughout the head and neck. So some high yield sticking points are going to be our eustachian tube, that is an endoderm structure, as well as our thymus and parathyroid glands, tonsils, and pharynx. So when it comes to our head and neck structures, most of them are going to be endoderm. However, it's important to know that our neural crest is what makes the bones of the head and face, and so that is not going to be lumped into this group of head and neck structures. To wrap up this video, remember that gastrulation is the event that creates our three germ layers: ectoderm, mesoderm, and endoderm. We also have that special function of neural crest cells, which are a special region of the ectoderm, and they create a high yield population of structures that test writers love to ask about on test day.